Thanks very much to the organizers for inviting me. It's my first time at this symposium, and I'm really enjoying hearing all the talk so far. I have no slide for conflicts of interest because I have no conflicts of interest. So that makes it really easy. Okay, so we're going to move out from the mucus layer, which is really important, and look at the overall physical barrier of the gut, because there's a lot of components that make this up. Gut motility, electrolyte secretion, mucus, epithelial cells, and the gut vascular barrier. And if we look at barrier function in its totality, it's made up of several different layers. Obviously, the outside layer, made up of commensal microbes, the mucus, which we just heard a lot about. Within the mucus, secretory IgA, antimicrobial peptides, the epithelial physical barrier. Underneath that, all of the immune cells, which make up the immunological barrier. And underneath this, the muscle layers and the vascular endothelium. So if we look now at the epithelium, movement across the epithelium is highly, highly regulated. So between epithelial cells, there are a couple of different pathways. One is called a pore pathway, and this can be changed when you change the kind of claudins and make actual pores that electrolytes can go through. There's also a leak pathway, which is slightly bigger, which can be increased by things like TNF, interferon. And then when cells die, there's ulcerations or apoptosis, you can get an unrestricted pathway, so passing right between the cells. And if you look at the junctional complexes, these are highly, highly dynamic structures. So the tight junctions, the adherent junctions, and the desmosomes, these things are regulated on a second-to-second basis. A lot of things can increase permeability, things like interferon, TNF, IL-1 beta, IL-4, IL-6, pathogenic invasive organisms, a lot of dietary compounds can act on the claudins, which are in here, can act on occlusion by phosphorylation. It can change the type of claudins that are in the pathway, so different claudins have different size pores. If you have a claudin 2, it has a very large pore size, a lot more can come through it. Whereas a lot of compounds also decrease permeability or ability of compounds to actually pass through this space. So things like IL-10, TGF-beta, EGF, short-chain fatty acids, we've heard a lot about. A lot of commensal microbes release certain metabolites that alter these pathways, and again, a lot of dietary compounds. Okay, now we've heard a lot that what you eat and what microbes you have will determine what metabolites are produced, both toxic and beneficial. And again, these metabolites go through the liver or via the lymphatic, and they go throughout the entire body. And again, fiber leads to enhanced barrier function, primarily through release of metabolites. So not only a trophic effect, but also in Treg development and as an anti-inflammatory, which we'll talk about. Okay, a little bit about the three main short-chain fatty acids. So acetate is absorbed through the gut. It alters neutrophils as it's going through. It has effects on epithelial cells. And it also goes systemically. It alters lung, it alters fat, it alters the brain, it alters the liver, it alters the pancreas, and bone marrow. So it has a lot of beneficial effects throughout the entire body. Propionate is primarily absorbed through the epithelium, again interacts with some GP-coupled receptors, but is primarily metabolized in the liver. Butyrate is primarily utilized within the epithelium, but it alters immune cells, induces T-regulatory cells. You've heard it is an energy source for colonocytes, and it also alters hormone release, but primarily in the epithelium. Okay, so why is gut barrier function important in human health? Okay, one of the main things is the fact that we have 1 trillion organisms here. We've got a lot of immune cells sitting here. These immune cells learn to tolerate these bugs. They know they're there, They know all about them. They learn about that in child development, but they tolerate them. And they maintain an immune tolerance, and they do not attack commensal organisms. And antigen entry and bacterial translocation are highly, highly controlled. You can get antigens coming across epithelial cells, being presented to the immune system, going through interperipheral lymph nodes. You can get antigens coming and interacting with dendritic cells. Again, it can go to mesenteric lymph nodes, or it can come in through Peyer's patches, interacting with immune cells there. But again, this is highly controlled interactions. When you disrupt tight junctions, when you lose your mucus, what happens is you get bacterial products coming across. You will activate dendritic cells that are sitting right there. You will get release of things like IL-12, IL-6, IL-23, IL-1 beta. You'll activate macrophages, they'll get all excited. You will drive T cells towards Th1 effector, Th2 cells, simply because bacteria and their products are not meant to be coming in an uncontrolled fashion. Whenever that happens, you will get a very excited immune system until the microbial products leave. And chronic intestinal inflammation, it can be induced by a lot of different, both exogenous and endogenous signals. So again, gut microbes, pathogens, they have a lot of microbe-associated molecular patterns, DNA, RNA, LPS. When these activate the immune cells, you get a primary response. And again, they will release things like IL-6, TNF. IL-6 TNF will act on epithelial cells to increase permeability. So whenever you have active inflammation, you will get an increase in gut permeability. You will then get cell death, which will release damage-associated molecular patterns, heat shock proteins, ATP, nucleic acid. These compounds will further drive inflammation, will further induce gut permeability. So it becomes a vicious cycle. And defective host containment of commensal bacteria can occur due to a lot of different mechanisms. So if you lose ability, mucus, you just heard about, if you alter expression of tight junction proteins, bacteria can come across. If you have decreased antimicrobial peptide secretion, you will not be able to contain bacteria and they'll be coming in. Or if the host cannot kill bacteria, because there's always going to be a few bacteria coming in, but they get killed and dealt with by macrophages. If you have a problem with macrophages and you can't kill them, then they're going to keep coming across. And when you have LPS coming across the permeability in the gut, it will go systemically, and you will drive subsequent systemic inflammation. And a lot of diseases are linked with a breakdown in gut barrier function. IBD, chronic kidney disease, we heard about, sepsis, pancreatitis, celiac, food allergies, alcoholic liver disease. All of these diseases have an increased gut permeability associated with the disease. But not only those, now we're seeing a lot of other diseases related to the brain, Parkinson's disease, alterations in gut barrier function, autism spectrum disorders, intestinal epithelial barrier disruptions, type 1 diabetes. All of these now are linked with altered gut permeability. Paper that just came out last month showing patients with type 2 diabetes now, again, this mucus barrier, they having bacteria that are encroaching into the epithelium. So coming way down, reduced mucus layer, driving host inflammation. But these are all correlation, association type of studies. How do you prove causation? We've heard about that. Mouse studies, it's easy. A lot of mouse studies of colitis, diabetes, you will see an increase in gut permeability before the onset of colitis. And you can prevent the onset of that colitis by changing the bacteria mainly. Using treatments to change the bacteria, to change gut permeability, prevent the onset of the disease in animals. But how do you study that in people? Is that true in humans? Does permeability increase before diabetes? Does it increase before IBD? And how do you study that? So back to the clinic. So this is an attempt to study in Crohn's patients. So this is a type of inflammatory bowel disease that has a genetic predisposition. So if you are a sibling of someone with Crohn's disease or you are an offspring, you have an increased chance of getting Crohn's disease, increased risk. So this is running out of Canada. A lot of European countries are contributing, as well as the United States. But we started this back in 2009. And we said, what if we follow offspring or siblings of patients with Crohn's disease and see who develops the disease? And then if we look at what is different about those people compared to the people who don't get disease, maybe we can find what are the factors that predispose to disease. So we have 5,000 subjects that we're following, of siblings and offspring, looking at intestinal barrier function. So we have assessed it when they come into the study. So they're healthy at this point, nothing wrong with them, no signs of bowel disease. Doing microbiota analysis, immune function, looking at all their environmental exposures, as well as a complete genetic breakdown. So now we're up to, I think we have about 60 patients who have developed Crohn's disease in this cohort. So this year we're going to be starting looking at what is different about those 60 individuals compared with the ones who did not develop Crohn's disease. This is the only way you can study this question in people. And you need large cohorts and long periods of time. Okay, what about normal aging? This affects all of us. What's happening? Okay, it is thought that it's an increasing inflammation that's a driver of age-related pathologies. Obviously, the food you eat, pathogen exposure, social environment, your lifestyle, all contribute. But it comes down to inflammation. And inflammation and gut permeability are linked. Gut inflammation directly increases gut permeability through cytokines and reactive oxygen metabolites. And gut inflammation also induces microbial dysbiosis. And this can then increase gut permeability. So why does inflammation always induce dysbiosis? Because of this. So when you have inflammation in the gut, epithelial cells will release oxygen metabolites, a little bit of nitric oxide. Neutrophils will come across, release some hydrogen peroxide. So now in the colon, you have an oxygenated environment. You got a lot of superoxide metabolites. So things like the anaerobes, the butyrate producers, are dead following this exposure. They can't handle these types of oxygen environments. But things like Enterobacteriaceae have enzymes that they can thrive on these components. So as soon as you have inflammation in the gut, increase in permeability, oxygen metabolites out here, you will get an outgrowth of pathobionts or Enterobacteriaceae in the proteobacteria. And you will lose your anaerobes. And when you lose your anaerobes, you lose your lactospiraceae, you lose your butyrate, you lose your propionate, you lose your metabolites that are pushing the production of T regulatory cells to an anti-inflammatory state. And what happens, now you have all the Enterobacteriaceae. These are more inflammatory type of organisms, and they are driving, they're invading, and they are driving a more inflammatory response. So our microbiome also changes with age. A lot of development in the early childhood. When you're an adult, it's relatively stable, depending on your diet. And when you start going into your elderly state, you start losing microbes again. You are losing your diversity. Things are going down. And it's interesting that this study just came out, and they were looking again at mice, but showing that it's the age-related changes in the microbiota that increase gut permeability in old mice. This actually resulted in a decrease, or associated with a decrease in innate immune function. So again, you now can't handle the microbes that are coming across. And if they did this in germ-free mice, there was no systemic inflammation. So it was a very nice study really showing that the age-related changes were related to gut permeability and changes in microbiota. Drosophila, very nice model system. Again, age-associated loss of barrier function was associated with altered metabolic immune signaling. And loss of barrier function was a better predictor of age-onset death than chronological age. So if you can keep your gut from getting permeable, you will have a much better quality of life in your old age. And I think we all care about that. This study, gut microbiota composition correlates with diet and health in the elderly. They looked at 178 seniors, both living in the community, residential care. Followed them as they went into residential care. I think we know that diet in residential homes is not that great. And they found that when they went into residential care, their diet changed dramatically, their microbiome changed dramatically, and their health changed dramatically. And that this is really a key modifiable risk factor in the elderly to help maintain a healthy microbiome and gut permeability. So really, aging is associated with the dysbiosis, along with the generation of enteric nervous system, increased permeability, leading to immune dysregulation, altered host metabolism, increased insulin resistance, chronic inflammation. One study, again, looked at prebiotic formulation in this population, 60 subjects in a nursing home. And again, the ones who, it was a parallel group, Indulin and Foss, and they had an improvement of hand grip and reduced fatigue. Didn't measure gut permeability or microbiota. Okay, what about gut-brain axis obesity and gut permeability? Okay, we know gut microbes can communicate with the brain in a lot of different ways through the release of metabolites like GABA, serotonin. Cytokines can act with the brain. Serotonin produced by enteroendocrine cells. And what happens when you're under stress, again, alters the enteric nervous system, increases gut permeability, getting that LPS, driving systemic inflammation, and showing that gut permeability is highly related to body weight, fatty liver disease, and insulin resistance in obese individuals, and that there is a clear link emerging between obesity and increased intestinal permeability, and that it's leading to inflammation, which leads to the problems of insulin resistance. So again, increase in adipocytes, secretion of adipokines, IL-6. This leads to an increase in gut permeability, chronic low-grade endotoxemia, and insulin resistance. And this activation of systemic inflammation can alter the brain, so you get activation of neuroinflammatory processes which lead to behavioral alterations and psychiatric symptoms, as well as mood disorders. So again, it's that antigenic and LPS driving from the gut that changes your brain through inflammation. So just to summarize, hope I've tried to convince you that gut barrier function is really key to homeostasis of the body, that it affects inflammatory tone, energy balance, gut motility, and appetite regulation. And again, consequences of a diet low in fiber, we've heard about these, but just to kind of put it all together. Reduce fiber, you select against T regulatory promoting bacteria. You start altering your gut mucus, you start altering your epithelial tight junctions. When you have high saturated fat, you increase your bile, you increase levels of sulfated compounds, you promote pathobionts, you increase hydrogen sulfide, which further reduces permeability, and you have this endotoxemia driving body inflammation. So going forward, using fiber to enhance barrier function has a really high potential for beneficial effects across the lifespan. To help with healthy aging, I think would be a key thing for fiber, have a role. It could help prevent disease in at-risk individuals, and which I didn't talk about, but it could help in treatment of active disease to address things like gut permeability and immune dysregulation. And I think couple of the main questions is that which fibers are most effective for which condition and what dosage, which we've heard about from other speakers. And also, if you're looking at the human trials, do you need to match the fiber that you're giving with what organisms they already have to try to drive those? And how do you actually do that in a human clinical trial? So I would just end there and say thank you very much.
So we welcome any questions for Karen.
Hannah Holscher, University of Illinois. Great presentation. I wholeheartedly agree that this is an important topic. Could you speak a little bit more to some of the tools that we should be using in markers within human populations? As an example, we know that LPS can be quite unreliable. It doesn't scale linearly. So something like LPS binding protein, or if you're doing mannitol, or some recommendations in that domain.
Yeah, so if you can, obviously doing something like a lactulose mannitol urine test, that's what we're doing for the GEM study. That's probably the best thing to do for monitoring intestinal permeability. And again, I agree that LPS in the serum is not a great marker. We've also looked at bacterial DNA in the serum, which again is good for a disease population, say inflammatory bowel disease, because they tend to have more. But if you look at a normal population, it's very, very low. So it's not that great of a measurement. So there isn't great if you, but the functional are the best if you can do that. Okay?
Thank you.
Bruce?
Hi, Bruce Hammerker from Purdue University. It was a fascinating talk. Thank you so much. I wonder if you might have a thought on, related to the regulatory side, if a biomarker there might be an inflammatory biomarker that is out there, or to think about, that would relate to, relate directly to the benefit of health side of things, and linking fibers, from the definition side, would particular fibers that would promote a low inflammatory state that would relate to health.
Yeah, which kind of comes back to the last question as well. So in our studies, we tend to measure IL-6 as an inflammatory marker, CRP as an inflammatory marker. We do measure LPS, and I try never to think about regulatory things because it just annoys me. So I just tend to think about the science. So we tend to go after the inflammatory cytokines as our biomarkers. Okay.
Go ahead.
Thank you for the presentation. I was wondering how the microbiomes affect the brain through the production of GABA, for example, in the gut. I know GABA is produced in the gut by the microorganisms, but how is that through inflammation reduction, or is it direct effect to the brain? How does it pass into the brain barrier?
Yeah, so there's a lot of ways that production of things like biogenic amines in the gut, serotonin, and those kind of compounds can affect brain function. Some of them will go directly into the brain. Some of the tryptophan metabolites will go directly into the brain. Some of them will alter cytokine production in the gut, and the cytokines will then go directly into the brain. And a lot of them will alter the vagus nerve activity, which will all, again, alter the brain. So there's a lot of different ways that these compounds actually, some of them will enter the brain, and some of them will just change nervous activity going into the brain. Okay.
Great. All right. Thank you very much, Karen.